Hello viewers, welcome to Methodology. In this video, I will try to give you an idea about the C and the N values of a cell at different stages of cell cycle. Let us first know what are the C and the N values. We use either small c or capital C to represent the DNA content in a cell, while small n or capital N to represent the number of complete set of chromosomes in the cell. This amount of DNA within a cell changes during large number of events that happen within the cell, like the process of fertilization, the DNA synthesis in the synthetic phase of interphase, and the M phase in the cell cycle. So, how does it change during these different phases? That is of importance in this video. In a haploid gamete, that is like the sperm cell or in the egg cell, the amount of DNA is just 1C because it's a single copy, and the number of chromosomes is haploid, or we can say 23 instead of 23 pairs. So it is 1N. But upon fertilization, when it goes to form the zygote, both the DNA content and the number of chromosomes in the diploid zygote doubles to 2C and 2N respectively, because it is the union of the contents from both the sperm and the ovum. Following DNA replication, the DNA content doubles again. This is what happens during the synthetic phase of interphase. Now, if it doubles from 2C, it will become 2 into 2C, which will be equivalent to 4C. But each pair of sister chromatids are still together, attached by the centromere. So, it is still counted as a single chromosome or replicated chromosome. So, the chromosome number remains unchanged at 2n. Now, if the cell undergoes mitosis, each daughter cell will return to the 2C and the 2N conditions, because it will receive half of the DNA content and one of each pair of the sister chromatids. So, cells at different stages of the cell cycle can also be distinguished by their DNA contents. For example, animal cells in G1 phase are diploid, containing two copies of each chromosome. So their DNA content is referred to as 2N. N designates the haploid condition. So the DNA content, as in the gamete cells of the genome, is N. During synthetic phase, replication increases the DNA content of the cell from 2N to 4N. So the cells in the synthetic phase have DNA content increased to 4N after replication. The DNA content then remains at 4n for cells in the G2 phases and also in the M phase of the cell cycle. But it decreases to 2n after cytokinesis. So, cellular DNA content can be determined by the incubation of cells with fluorescent dyes that bind to the DNA, followed by an analysis of the fluorescence intensity of the individual cells in a flow cytometer or in the fluorescence activity with the help of a cell sorter, which can distinguish the cells in the G1, S, G2, or the M phases of the cell cycle. Thank you for watching. I hope you liked my video. Please do not forget to give a thumbs up, share it with your friends who may find it useful. Also, do not forget to subscribe to my channel. Thank you once again.